dear colleagues, I show you the results of our work and guide you through my poster secondary structure of the SARS-CoV-2 genome affects molecular evolution. You can see basic description, hypothesis, and main results on the poster that's fixed on the slide. Our hypothesis is that genomic secondary structure of SARS-CoV-2 might be under selection and play a certain role in the evolution of the coronavirus. We are studying how secondary structure influences the mutagenesis and selection directions. To do that, we use SARS-CoV-2 genomes from the GSA database and build it to a genetic tree with reconstructed ancestors. From the tree, we release substitutions and use them for our analysis. Let's see our results. Firstly, we check the average number of substitutions per position in 20 nucleotides genomic intervals. We can see that in structured stem loop regions, orange, there are significantly less number of mutations than in unstructured regions. We think that it may be consequence of negative selection because of stem loop structures have many functions in SARS-CoV-2 processes. Also, reduced mutagenesis can be caused by physical barrier of stems for some mutagens. Another result is the mutational spectrum of the coronavirus. We calculated the mutational spectrum at native for 4 degenerate sites, taking into account their pairing. And we used only mutations from the terminal branches of the phylogenetic tree to reduce selection impact and see most natural mutagenesis. The common substitutions are cytosine to uracil, C2U, and guanine to uracil, G2U, which is consistent with the results of other studies. Presumably, C2U is caused by deamination by cell deaminases ApoB, which are responsible for antiviral cell protection. It's considered that G2U is a signature of the oxidative stress. We can see that in paired sites, rate of substitution C2U significantly decreased. Dark gray column is lower. But rate of G2U is similar in paired and unpaired positions. We assume that this can be another evidence that substitution C2U is in the coronavirus caused by deaminases ApoB, which works only on single-stranded RNA or DNA. Also, nucleotide states, paired or unpaired, have no influence on reactive oxygen species, as expected. To study selection directions, we calculated distances between closest mutations with different selection impact. We know that mutations from terminal nodes of the phylogenetic tree are least influenced by selection. Terminal mutations appeared largely because of natural mutagenesis. We use these mutations to calculate the mutational spectrum on the previous slide, but mutations from the root of phylogenetic tree are maintained only by the selection. Because of that, we split the substitutions to subsamples from 3 branches: terminal substitutions S1, pre-terminal substitutions S2, and so on to the root. In each substitution sample, we calculated distance between closest substitutions. See figure 4. We can see that for the first 3 samples, for structured and unstructured regions, there are no significant differences. They are equal. But for other samples in structured regions, orange bars, we observe less distance between closest mutations. Let's look at sample 4 in detail, these bars. The distributions strongly overlap, but differ statistically significantly. In the area where distance equal to about 200 nucleotides, this area, it can be seen that structured regions prevail. Let's take a look at their distribution functions to study their relationship. Let us divide these lines is distribution functions. Let's divide the distribution functions of structured regions, orange line, to CDF of unstructured ones, blue one. Figure C is division. We observe a region of 200 to 400 nucleotides where structural regions predominate. This means that mutations in structured regions are more likely to occur 200, 400 nucleotides from another mutation. We associate this observation with periodic structure of SARS-CoV-2 genome. To find selection impact to this effect, we checked other substitution samples. And we have seen that this effect exists on different selection impact level. On substitution from terminals and pre-terminals of phylogenetic tree branches, there is no effect. It's dashed lines. But on basal branches, there are nearby the root, the effect is only getting stronger. Here we propose the SARS-CoV-2 genome. In SARS-CoV-2 genome, there is a periodicity that determines the distance between substitutions, and which is supported by the forces of selection. We suppose that this periodicity is determined by stem loop structures. Yes, and thank you for your attention.